The overall goal of the following experiment is to determine genomic binding sites of bacterial two-component response regulators using an in vitro microarray-based assay. This is achieved by cloning and purifying a tagged response regulator protein, activating it by in vitro phosphorylation, and determining one gene target using electrophoretic mobility shift assays to use as a positive control for the downstream microarray steps. As a second step, the purified response regulator protein is mixed with shared genomic DNA, which allows the protein-bound DNA to be separated from input DNA using affinity purification. Next, the protein-bound DNA and input DNA are amplified, qPCR verified for the enrichment of the positive control gene target in the protein-bound fraction, fluorescently labeled, pooled, and hybridized to a tiling microarray in order to determine the genomic binding sites of the response regulator. The results show the most likely genes that are regulated by the response regulator under study, and the results can be further validated using electrophoretic mobility shift assays.
The main advantage of this technique over other methods like the in vivo chip is that it can be used even if the activating signals and the conditions are not known for the two-component systems.
This method is of great importance to the bacterial signaling field, as the vast majority of response regulators have unknown gene targets, and this method helps to unravel intricate regulatory networks.
Demonstrating this procedure will be Amy Chen, a research assistant from my laboratory.